We're going to talk about gene expression or DNA expression, so Roman numeral 3 on your outline. So let's talk about a gene, right? Genes are going to be these inherited units on the DNA molecule, and we have, anywhere between 30 and 35,000 genes. So let's take a look at a gene, but before that, just what does genetic expression mean, or gene expression? So how do you go from a molecule of DNA, right, that we looked at and we know the structure now, but how does it really translate into a creature, right, into this fully formed puppy? So it turns out that there's different steps along the way, and the big secret is that DNA is a code, and it's coding for a protein. So at the end product of your genes, so if you're asked this on a test question, like what do your genes code for? They actually code for proteins. And those proteins are going to basically be little machines that work to build your body and to do the chemical reactions that you require. So if you remember, enzymes are proteins, and enzymes are critical for all of our chemical reactions that happen inside our cells. If you think about what your body is made of, your hair, your skin, your nails, your muscle, it's all protein, right? It's proteins, and then there's enzymes that create other molecules. So there's two steps to genetic expression. The first step is called transcription, and we're going to transcribe the information on DNA into RNA, and then the RNA is going to leave the nucleus and go into the cytoplasm where it's translated. So that second step is called translation, and it's going to build an amino acid chain, right? So we know this is the first step in making a protein. This is called the primary structure of a protein, and then we're going to make our protein. So one of the things I like to talk about is that, before we move on, is that your body, right, the trillions of cells that make up your body, all have the same DNA. And so why is it that your skin cells behave a certain way, and your neurons look and behave a certain way, and your fat cells and muscle cells look and behave completely differently, right? They all have the same instruction set. So what they're doing is they're expressing different genes. So you can think of your genome, right, all the DNA in every single one of your cells, as a library. And then if you are a neuron, you wanted to conduct electrical impulses, then you're going to access only the information you need to do your job, right? To build the machinery to conduct electrical impulses, whereas a fat cell is not going to do that, right? A fat cell is not going to access the nerve genes or the neuron genes. They're going to access other genes in that same library. So every single one of your cells has the information to be anything, but it doesn't, because they specialize and they access or express only the genes that are required for their specialty. So hopefully that makes sense to you. And so now let's go ahead and look at what is a gene. Oh, so I forgot I had this slide in here. So this is just the steps in picture form. So there's our DNA inside our nucleus. This is our double-stranded DNA molecule. We're going to transcribe, right? So that first step is called transcription. The information on DNA, we're going to put it into a molecule of RNA. Notice that the molecule of RNA is much smaller, and it's also single-stranded because we're only accessing the information on a gene. So a gene is going to be a small little segment of DNA. The RNA molecule will then exit the nucleus, enter the cytoplasm, because where does protein get made? Protein gets made on ribosomes, and ribosomes are in the cytoplasm. So the RNA comes out, and here's our protein being made in a process called translation. All right, so concept check. DNA is what to RNA? And the answer is A, transcribed. Okay, so let's look at the picture here we have of transcription. Actually, let me just show you this one first, because in your notes we talk about a gene, letter A, a gene, and let's look at the important areas of a gene. So promoter region, coding region, and terminator, which is here. So this is our DNA molecule, and just a little portion of it. And there's a very special region of a gene called the promoter DNA, or the promoter region. So this is kind of like the go area of a gene. Everything in the middle here between the green and the red, this stuff here in light blue, this is going to be the content of your gene. So let's say this is a gene for myosin, which is a protein that's found in your muscles. And this is the information to make that protein, okay? That's called the coding sequence, or the coding region. So how does the machinery, in this case an enzyme called RNA polymerase, know where myosin starts and when myosin ends? It doesn't, right? So it has to, the DNA has built in these little start areas. So it's or what we call it upstream. So this is upstream from the gene. So the RNA polymerase will bind at the promoter region and get ready to start the transcription process. And it'll transcribe, so you can see the steps, right? The initiation process. We're going to start to transcribe the gene. So what this RNA polymerase does is it opens up those two strands of DNA, as you can see in this picture, and it starts to, just like when DNA is copied, it will use one of the strands of DNA as a template, and it will read the template, and then it'll add the nucleotides on to make RNA. And it continues to do that, right? It unwinds as it goes to make this piece of RNA. And then it stops by hitting this termination sequence, also known as the terminator. So it's the stop. And so this way, this molecule only transcribes the information that makes that myosin protein, right? It's not going to go before and get useless information before. It's not going to go after and get extra information after. It's just going to do what the DNA tells it to do between the promoter region and the terminator region. Okay? So let's take a look at this animation, see if we can